we go further to nucleic acid extraction, I just want to remind us what our nucleic acid is. We have our DNA and our RNA, and then the structure. We have the linear nucleotide that is linked by phosphodiester bond. We have nucleotide that comprises of three components: our nitrogenous bases, our carbon sugar, which is deoxyribose in DNA and ribose in RNA, and then our phosphate group. So this is just to remind us of the structure of our nucleic acid, so we don't forget that. So this is RNA and this is DNA. So this is the physical structure of how our nucleic acid looks like. So go to nucleic acid extraction. So what is nucleic acid extraction? Nucleic acid extraction is the process of isolating your RNA or your DNA from your biological materials, your biological samples, and this ranges from your hair, your saliva, your blood, your tissue, and any other biological samples you can think of. Some people, there was a time I was talking to someone that, nucleic acid extraction is the foundation of all downstream molecular applications that you can do. The person was like, nucleic acid extraction is just there. I was like, okay, imagine you want to perform PCR, or even you want to do forensic investigation. You will still have to extract nucleic acid from your host. So what if while extracting your nucleic acid there was a mistake or anything? Then it's going to affect whatever it is that you intend to do with the project. So that's why whenever I'm trying to introduce or trying to write my skills, nucleic acid extraction technique is the number one on the list. I make sure that, because in as much as it looks simple, it is still very important. In fact, it is the most important techniques in molecular biology. So it's very crucial for accuracy of downstream molecular applications such as your restriction digestion. So restriction digestion is when you use restriction enzymes to recognize and also to cut specific sequences. So before you can do that, you also need to extract your DNA, and it requires high quality and intact DNA. Also for PCR, that's polymerase chain reaction. Let's say you want to use your conventional PCR, that's the standard PCR, or your reverse transcriptase, that's when you want to convert your RNA to cDNA, or your qPCR. Let's say you want to do, that's real time PCR for gene expression studies. You also need your DNA or your RNA for that as well. Your microarray analysis, you need intact and high quality nucleic acid as well for you to hybridize the probes on the chips. We know that microarray is a chip that is used in doing that. Also for your nucleotide sequencing, your phylogenetic research, your forensic investigation as well, your DNA extraction from your maybe your blood, saliva, hair, or tissue is the first step in forensic investigation. Also for cloning, you need your nucleic acid extract. So you see, there is no molecular applications you can think of that you don't need nucleic acid extraction for. And when we say nucleic acid, you are not just extracting for extracting sake. You need high quality DNA and RNA, and that's what will take us to our next topic, which is nucleic acid quantification. So when we get there, we'll understand why we need high quality nucleic acid. So for extraction method, when you want to extract, there are two ways to extract. No matter how you want to do it, just two ways. It's either you use an in-house protocol or use a commercial kit based. In-house protocol simply means that you put this thing together yourself, and kit based means that, you get to buy the kits from the manufacturers. Let's say you buy from Nordgene, you buy from Gene, you buy from Gene Aid, you buy from Imo, and the likes. Please, can we still hear me? And can we see my screen?
Yes, yes.
Okay, good. The difference in the two is that for in-house protocol, it is transparent, it is modular. That means you can modify it. You can say, for sample one, let me use 200 microliter guanidine outside it. Sample two, let me just tweak a bit. Let's use 150 microliter of guanidine outside it. It is adaptive. It's less expensive, but it's time consuming. Instead, it's not like kit based is very fast. Kit based, when you want to extract 10 samples, it depends on your know-how as well. But at least even for a beginner, if you want to extract say 10 samples, it should not take you less than an hour if you really know what you are doing. But the only thing that consumes time in kit based is the centrifugation. You centrifuge a lot when you are what's it called, when you use commercial kit. Then for the in-house protocol, it is less reliable. Limit to the downstream applications you can use your in-house protocol for. You can only use it for standard PCR. That's the conventional PCR. I don't want to do cloning now. Like, oh, I want to add my DNA, but I want to use the in-house protocol. I just want to waste my time, my money, my resources, because it's not going to come to it, because the yield is not as great as the kit based. There is no quality guarantee. And you also need to know the principles behind extraction before you can use in-house protocols. Sorry. Before you can use your in-house protocol. So that's the difference between the two. So these are just the example of the main. Let's say you are doing blood, you can get for blood. Let's you want to do for tissue, there is kit for tissue. If it's soil, you want to extract your DNA from your soil, there is what's it called? There is kit for that, for yeast and any biological sample you can think of. So whether you are doing we are now at principles of extraction. Whether you are doing your in protocol or your kit based, these are the principles of extraction. That was why I said you need to know how extraction works before you can do in-house protocol. Let's say for in-house for your extraction now, the principles of extraction, you follow your lysis, you break down the membrane before you can get to the nucleic acid material, the genetic material, whether your DNA or your RNA. Then you precipitate, then you wash, then you elute. So let's say you don't know how the principles behind the extraction now. In your in-house, you now started with precipitation. You have started the whole work. So you have to know the principle that okay, cell lysis first, then precipitation or purification follows, then I wash, and then I elute before I now store my extracted nucleic acid. So we are going to talk about each of these principles and their types. So this is just a pictorial representation of the principles. Although when you're using a commercial kit, there is a protocol comes with it. Every manufacturer has their own protocol attached to each kit that you buy, but it still follows the same principle. Whether you buy from Zymo or you buy from Creagene or you're going to lance first, you press it, you wash, and then you elute before you store. Lysis. That's the first stage we are doing. So there's going to be a lysis buffer. Usually contains two lysis buffer. Yes, yes, two lysis. You have the GB lysis buffer, and then you have another lysis buffer. So this lysis buffer, you to prepare anything yourself for commercial kit. Everything is prepared, but whatever it is that they need you to add to the already prepared reagent, they will indicate it on the bottle. Example, when you get to your wash buffer, they will ask you to add 100 ml of ethanol, 100% ethanol to the already prepared wash buffer. But for lysis buffer, you don't add anything. It's cell lysis. So I said we should imagine ourselves now that we want to extract blood sample. So we already take our 50 microliter of our blood inside a 2 ml microcentrifuge tube, and then we are using gene age genomic DNA kit. So the first thing they will ask us to do is to take the blood, that volume of blood inside a tube. Then it will follow by adding a GB lysis buffer. So this lysis buffer contains this cheotropic salt, that's guanidine thiocyanate or guanidine hydrochloride. This lysis buffer will do is to destabilize the hydrogen bonds and then all the interactions within that genetic material. And also sometimes a detergent is often present in the lysis buffer as well, but it's not common. Like for Zymo now, Zymo doesn't add detergent inside their lysis buffer, but the rest of the manufacturer that I have used, there is detergent. So whenever you put your lysis buffer inside your sample, whether it is blood, whether it is tissue, it tends to, become soapy. It will look as if you added soap inside of it. So that is for lysis buffer. Let's still continue that mental image. So we've added our lysis buffer now. Then we have three types of lysis buffer. We have the three types of cell lysis: the physical disruption, the chemical lysis, and the enzymatic lysis. So for the physical lysis now, you can lyse by freezing or thawing. If you are using this physical lysis now, is you using your in-house protocol. So I just put this here for the sake of people that want to, that might want to do in-house protocol. So you freeze and thaw, or you grind in liquid, you grind in liquid nitrogen, or you do ultrasonication. Ultrasonication is not really something we do here in Nigeria. At least all the labs I know that I've worked with, we usually don't. Whenever we want to do in-house protocol, we usually don't do ultrasonication. Bead milling, the most common one is the freezing, the grinding in liquid nitrogen, the bead milling. So the bead milling is, there is a tube that has beads. So there are beads inside of it. So we now shake that beads. We add the sample inside the tube that contains the beads, and then we start shaking. So we do this bead milling for, let's say, tissue samples. So you can use the bead milling, and then boiling as well. So we boil using the heat block, that's the water bath. We can use the water, well, that's how we boil, to also disrupt the cell membranes and cause cell lysis as well. Then for this chemical lysis, this is when you make use of your um commercial kits. So it contains this ionic detergent, and then some kits also has, I said earlier that we have two types of lysis buffer. So the second type is the non-ionic solvent that comes with the kit as well. So that one contains butanol. The first one contains SDS, guanidine thiocyanate, guanidine HCl, and this will inhibit the enzymes and also going to interact strongly with the lipids and also denature the protein. Sorry. And we also have special buffers. That's part of the chemical lysis as well. Then we have enzymatic lysis. This is when we make use of enzyme to also lyse, to break down the membrane. So we have the proteinase K, we have lysozyme. If you want to extract a tissue sample, there is there are some kits that will tell you that, okay, there is lysozyme inside of it. Some don't put lysozyme because lysozyme is expensive. But if you want a high yield and a good quality DNA, you need to add lysozyme to it. Then we go to the next principle. Principle one was cell lysis, and we've talked about the types of lysis that we have. So now after we have lysed, we have centrifuged, we have followed the manufacturer's protocol. Then we move to the next stage, which is purification or precipitation. So it is at this stage where the nucleic acid now bind to the column. So after you have spinned down your your what's it called, the blood sample with the lysis buffer. You already have your pellet and your supernatant. Then you transfer into a new collection tube. But instead of that using the first collection tube you use, you're now going to use a GD column tube or a filter column or a spin column. So this column has silica gel. It has silica. So the column is, you can remove the column. The column is attached to a collection tube. So whatever you put, it's just like a sieve. Whatever you put at the top, when you centrifuge, the supernatant will go to the bottom. Then you can discard that flow through. Then your nucleic acid will now bind to the silica, the column at the middle. So that's what happened at precipitation. And so, and also when you precipitate, there you add alcohol. Sometimes they ask us to add isopropanol. It depends on the manufacturer, but either alcohol or isopropanol works well. So this will further enhance and influence the binding of the nucleic acid to the silica. And also the percentage and the volume of your what's ethanol are very important. Some manufacturer will say, oh, add absolute ethanol, that's 100%. Some will say add 70% ethanol. Some will say add 400 microliter. Some will say add 600 microliter. So it depends. You have to that's why when you are when you are extracting your nucleic acid, your mind has to be where you are in what you are doing. Because if they say you should add 500 and you add, even if it's as 510, even if it's 505, you already, disrupt the whole workflow. Too much of ethanol will bring down the degraded material, and then that's going to affect your reading when you want to quantify to see the yield and the concentration and the impurity of your DNA. And then when it's too little as well, when you add small amount of ethanol as well, it will impede the washing of the salt from the membrane. You will not wash off the salt. It's also going to affect your readings as well. So we have, can we all see hear me, Christiana?
Yes, you can hear you loud and clear.
Yes, I can
Okay,
hear
good.
you.
Okay, good. Thank you very much. So for the purification now, we have the chemical purification and the alcohol uh purification or precipitation. So we make use of the phenol, chloroform, isoamyl, and the likes. Like I said, it depends on the manufacturer. When you are extracting your DNA, you cannot what's it called, put the whole protocol, step by step protocol into your head, no matter how brilliant you are, no matter how good you are. But the only thing you can do is know the principle. Know that, okay, whenever I get my kit and I want to extract, I know what I'm doing first. I'm lysing first. So no matter if you are even new, maybe you have been working with Zymo kit before, and then you find yourself somewhere that they are using a Norgine kit or Forgine, you will not even be surprised, because you know that, okay, no matter what, I know that I lyse first, then I precipitate. So even if the kit is saying that, oh, they are using phenol for their own precipitation, and then the Zymo uses chloroform, you know that you are still on track. You are still on track. You know what you are doing. And for alcohol precipitation, two. We use ethanol, sodium chloride. For the cationic salt, ammonium acetate for the purification or the precipitation. So this stage, we have removed the protein. Our nucleic acid have what's it called, are binded to our silica, that's to our column. Then we now want to wash. Yes, we now have our nucleic acid. So imagine now you have spin down your what's it called, you have spin down your, you have purified now. Your what's it called now is now in your nucleic acid, whether your DNA or your RNA is now in that silica. Then they will ask you to transfer, to discard that flow through collection tube and insert a new one so you don't contaminate it. So now we now want to wash. We cannot just use our DNA like that. There are still some salts, there are still some impurities that are still bound to the column. So for us to wash, we use a wash buffer. And most kits, you wash twice. It's just as how we wash our clothes. When we wash with soap, we will rinse again, to make sure that there are no soap, that are still left on the cloth. So after centrifuging your latte, so let's still continue that image. We are now centrifuging now. We've added our wash buffer. Usually you add 600 microliter of your wash buffer. Most kids they write wash buffer or W1 buffer and then W2 buffer. Some will write W1 buffer, then wash buffer. And this wash buffer, you usually add uh ethanol to it. Usually absolute ethanol. That's 100%. So they ask you to add 100 ml of ethanol to the already what's it called, made wash buffer that is in the container. Always, if they have added it, they will indicate that they've added it. In fact, when you open your wash buffer, it smells like, if you are very familiar with the with the scent of alcohol, immediately you open your wash buffer, everywhere starts smelling like ethanol. So the wash steps will remove what's it called, impurities, protein, or your salt or your phenol residues that might still be binding to your nucleic acid and the silica. So you wash twice and then you wash again. So this is very critical. Sometimes when we are done washing, we make sure that we wash. Even sometimes when you're working with let's say animal tissue, it's advisable you wash twice, because animal tissue sometimes their DNA can act funny. And even when you wash twice, no matter how good you are, you can still, when you check your concentration, you can still get a low yield. So some kits usually recommend 2 ethanol washes. I think yeah, this that should be forging. Forging uses 3 washes. You wash twice, but the third wash is just going to be the wash buffer with little amount of ethanol inside of it. So now we have washed now. We have centrifuge. You centrifuge at 14,000 RPM for 30 seconds. So we have done that as well. We've done wash 1, we've done wash 2. Are we following? Now we want to elute. After we have washed now, we will discard the collection tube. But
Yes, yeah.
the would okay, thank you. So now we want to elute. After we have washed now, we will discard the collection tube with the flow through. That our column, we will now place it inside a new maybe 1.5 ml microcentrifuge tube. That's where our DNA or our RNA will be eluted inside. So we'll now add our elution buffer. The elution buffer also comes with the kit. So the elution buffer, you add 50 microliter in most cases. But there are cases that you might later need more than 50 microliter for your work. So some kits, they usually say we should elute 100 microliters. So there is no assay you want to do. Even if you want to do all the molecular downstream application in this world, that your 100 microliter will be enough. Yeah, but if you are very certain that, okay, I'm just using this for just these applications, then you can do 50 microliter reaction. So when you take your 50 microliter of your elution buffer, or you take 100 microliter of your elution buffer, then you place it. I hope we are with our pipette. Then you place it at the middle of that filter column, because if you put it on the wall of the tube, you are not going to elute your DNA. The DNA or your RNA is at the middle of that column. So you have to make sure that that elution buffer also goes to the center. So once you dispense the 100 microliter elution buffer, then you let it stand for 1 minute. Once you let it stand for 1 minute, then you now centrifuge for another 1 minute. By the time you are done centrifuging, your DNA is now in that new centrifuge tube, that microcentrifuge tube. You can now discard the column. But before you discard the column, we always make sure that we quantify first. If you discard it immediately, your spin column, and then you realize that you have a concentration error, then there's nothing you can do. That's why whenever we finish, we get a paper towel, we just place the spin column then, and after we check our concentration and we see that it is good, we can now discard them. So that is for elution. So we are done with our extraction. So when you are done with your DNA or your RNA, you have to store them immediately. DNA is even in the lab, we call DNA like we call DNA a tug. There's nothing that you do to DNA that DNA cannot. It can withstand any situation. But you see RNA is a princess to the core. Even when you are extracting your RNA, you have to extract it on ice block. Like you have to place ice and then do it everything you want to do, even to your tube. Your tube must be RNA free, because RNAs, when you're extracting your RNA, it's advisable you don't talk, or is it that you wear a nose mask, because even as little as your saliva or anything, it will degrade. RNA doesn't have, it has a very short lifespan. So you have to treat RNA with care. And that's because of their structure. We know that RNA is single strand and then DNA is double strand. So immediately you extract your RNA, you take it to -20 or 4 degrees Celsius or -80. Even DNA, you can still put DNA in the freezer, in the fridge. But RNA, if you put your RNA in the fridge and not the freezer, you cannot use more than 1 hour. The RNA will degrade. But if you are using them, if you just want to, okay, let's say we just finished our RNA and our DNA now, and we want to use them next month. So we have to put them in the freezer, maybe 4 degrees Celsius, -20, -80. That's how we store. So this is for DNA. Your lysis buffer is either STS, EDTA, your protein digestion, that's for your purification, and this for phenol as well. Then also precipitate with ethanol or sodium acetate. All these things, you cannot use it for RNA. For RNA, can we still hear me?
Yes, we can.
Okay, good. Thank you. For RNA now, the lysis buffer in RNA for RNA extraction, guanidine thiocyanate or B-mercaptoethanol. And when you're using B-mercaptoethanol, it's a bit I won't say carcinogenic like that, but it's not really, I'm not going to say it's not safe, but it's still safe. When you want to use it, it's best to use it in as an environment that is not open, that everyone can. That's why when we want to extract, we have an extraction room that it doesn't get to the air like that, because number one, the smell, and you cannot what's it called, perceive it for a long time. So B-mercaptoethanol for RNA extraction, your phenol and chloroform as well. And then precipitation is isopropanol for RNA. For DNA is alcohol. And then you wash for RNA, you wash with 70%. You cannot wash more. You cannot wash with 100% with RNA. You already wash all your RNA. Like I said, RNA needs to be treated like a queen. But for DNA, you can even wash with 200% ethanol. You still see it there. And then you must always do a DNA treatment for RNA. Although some kits will say it's optional, but I will advise that you do that when you want to extract your RNA. So these are the types of nucleic acids that we extract. The gDNA, that's for that's the old DNA. Like when you extract your DNA like this, that's your gDNA without doing anything there. So you can use it for your whole genome studies, your thing. If you want to do NGS, that's next generation sequencing, and then genotyping. We also have plasmid DNA, so we use this one for cloning and transfection. We have the total RNA, we use this one in transcriptomics, and then in qPCR, that's real-time PCR. Then mRNA for gene expression. We can use total RNA for gene expression as well, this small RNA for non-coding, and then the viral RNA and DNA for pathogen detection. So all these nucleic acid, all these extracted nucleic acid, there are kits that you can use for the, so it depends on the study. You cannot want to do cloning now and say you will use a whole genome DNA, or I want to do qPCR now and I want to use genome DNA. It's not possible because qPCR for expression study you have to use RNA, then you now convert it to a cDNA before you now run it on qPCR machine. So the applications of nucleic acid extraction, I already said like you need it in most downstream molecular application for research, for diagnostics, for forensic, and also for parental prenatal testing, agricultural genomics, everything that you can think of. Nucleic acid extraction is the foundation. It's what you build on, and that's why the nucleic acid extraction must be very efficient. So whenever you do that, you know that okay, I'm certain that, so when you encounter any problem, you'll be certain that okay, this problem is not coming from my extraction. Oh, it's either maybe when I was running my PCR, maybe the temperature was not evenly distributed. You will know that the issue is not from your nucleic acid. So you have to make sure that you are very good when extracting and when extracting your DNA, even if you are good, you have to learn speed as well. Because let's say you want to extract RNA, you cannot use 2 hours extracting RNA. Even before you are done, the RNA will have degraded. You are just wasting your time. So you have to learn speed as well. But while you are learning speed, you also have to, what you are doing. So you won't mistake watch buffer for elution buffer. You also have to know that as well. So this is just so when we extract, even if you are very good, you are a pro at this, we still encounter some problems, and these are the possible solution. So when the problem is low yield, like I said, if you don't want to start from the beginning, because this optimized lysis, that means you are starting from the beginning again. Your first go to should be your elution. We elute, like I explained earlier. You will elute to see that okay, maybe some of my DNA, they still, they refuse to come down. Maybe they are still bound to the column. Let me just re-elute. So once you do that, and then you still have what's it called, low yield, then it's advisable that you start again. Then you optimize your lysis. So if you use 400 microliter before, then you can now do 450. Then if the problem is contamination, you have to include extra wash steps, or you improve your phase separation. Maybe when you were supposed to use a filter column, you used a GD column. So most time too, those mistakes can also cause you your concentration of your nucleic acid. If it's RNA degradation, number one, you have to ensure that you have to ask yourself that did I use RNA free tech what's it called, equipment? And if it's PCR inhibition, ensure complete ethanol remover. So maybe after you have run your PCR, the bands were not as great as it should be, then you have to start again. Make sure that you get it well. So conclusion, extraction quality is key to molecular biology success. The study you want, oh, I want to test for this, I want to see what is responsible for this, oh, I just test this. Your extraction quality matters, and you have to choose method based on sample and application. You cannot use a kit that is meant for plants to extract blood. It's not the same. Even if the principles are the same, the reagents are not the same. And you have to use proper quality checks for reliable data. And that takes us to our next topic, which is nucleic acid quantification. So these are the outlines we are going to talk about. the quantification methods that we have, and we'll compare the methods. So let's get into it. So what is nucleic acid quantification? This is the measurement of your DNA or your RNA concentration and purity. And this is very essential before you send that nucleic acid for any downstream application. Before your PCR, your sequencing, your cloning, you have to make sure that you check the concentration and the purity. It doesn't take time, but at the same time it is a very crucial part you should not miss when you are in the lab. And this is why it is important. When you when you quantify your DNA now, and then you know that you have a very good yield, very good impurity, very good quality as well. When there is now a problem with your PCR, you know that you are not going to feel like, oh, maybe the reason is with my DNA. Oh, maybe my RNA is bad. You will know that that's not the issue because you already quantify, you already passed that check. And it also, prevents failed reaction. You know that, oh, I should not use this, my nucleic acid. Let me re-elute, or let me start again. Because if I should use this, I'm just going to waste my time. It also helps you to identify your sample contamination. Do I still have protein in my sample? Do I still have DNA? Do I still have RNA? Do I still have phenol? Do I still have salt in my sample? So we have three methods of our nucleic acid extraction. We have UV spectrophotometry, that's nanodrop. We have the fluorometry, that's we have the qubit fluorometer, we have the picograin, and then we also use agarose gel electrophoresis. We'll start with the nanodrop. So for nanodrop now, this is how a nanodrop looks like. You see that place that has that black wire, that's where you open it. So when you open it, there is a pedestal there. This pedestal at the bottom, the one that is the cover is resting on. So that's the pedestal where you add your DNA or your RNA. You take 2 microliter. Most nanodrops are calibrated to 2 microliter. So you only take 2 microliter of your sample, then you place it on the pedestal, and then you close it back. Immediately you close it, you connect this nanodrop. The software must be on a desktop or a laptop. So immediately you put your DNA or your RNA on it, or even your protein, and then you just click measure on the laptop, and then it measures, and then you see your result. There is a picture of how the results look like. When we get there, we are going to see. So it is used to accurately measure DNA concentration, requiring only 1 to 2 microliter of your sample. Like I said, the nanodrop is calibrated to take 2 microliter, some 1 microliter. So it ranges from 1 to 2. And this nanodrop, it measures it is based on the absorbance of light. So that is, it measures how much UV light is absorbed by your sample at a specific wavelength. So we have the 260 and the and also the 260 230. So nucleic acid, DNA and RNA, they absorb maximally at 260 nm because of the aromatic basis. But for proteins and other but proteins and salts, phenols, and other contaminants, they absorb UV light at different wavelength. So now our 260-280 ratio is used to assess protein contamination. That is, your ratio of absorbance at 260 nm, that's the nucleic acid, to absorbance of 280 nm, that's your protein, that's your 260-280. And the typical values is greater than or equal to For DNA, approximately 1.8 to 2.0. For RNA is 2.0. Let me Then for 260, 230 is the ratio of absorbance at 260 nm to absorbance at 360 nm. So this one is used to access for other contaminants like phenol. When we get to the result, I'm going to interpret the result to you, and then you are going to understand better. For nanodrop, there is no dye required, unlike fluorometer. Fluorometer requires a dye. You have to add dye to the sample before you can use the qubit fluorometer. This is how a fluorometer looks like. So the fluorometer has a manual, and then these wells that you can see, that's where you place your tube. So they ask you to take 5 microliter or sometimes 10 microliter of your DNA sample, and then you put it inside the tube, and then you add the buffer and the dye to make it 100. That's 90, and then you place it. You take 190 of that. You make a mixture of your dye and your buffer, 190, then 10 microliter of your DNA or RNA to make it 200. If you are taking 5 microliter, that means you have to make the mixture 195. Then you put it there. Then you will select if you are doing DNA, RNA, protein. Then click on it, and then it measures and it brings out your values for you. It is highly specific and is sensitive. Unlike uh nanodrop, that is going to tell you if there are still contaminant, or you still have salt, or you still have this. This one is not affected by contaminant. That's why it's highly sensitive. Once you put it, it's just going to show you the yield and the concentration. So if you are good to work with the concentration, then you can continue with other applications you want to use the DNA for. Then for agarose gel electrophoresis, while nanodrop and fluorometer can tell us the concentration and the yield, they cannot tell us the integrity. That is where agarose gel electrophoresis comes in. This tells us the integrity. You can see the first three, they look very what's it called? They are not as deep as the second three. They are another set of three, the EGZ, HIE, L, and G. Then the next set too, they are also faint. Then the next set, then the next set. So what this means now is that whatever I use, these three sets that are very bold, whatever downstream applications I use them for, I would definitely get a good result, no matter what. Even if that's my first time entering a lab, I would definitely get a good result. For those ones that are faint, it's not as if I'm not going to get a result, but their integrity is not as high as this. So gel electrophoresis, checks integrity of the nucleic acid, and you compare it. You make comparison with the DNA ladder. Your DNA is not supposed to move because you have not done anything to it. It's still a old DNA. It's not as if you have amplified. It does not undergo anything. So when you put your DNA ladder here, this is a 20,000 base pair ladder now. If now, let's now assume one of the DNA now move to, after running with power, after you use electricity to run it, you plug it to your power pack, your gel and whatever, and then one of the, let's say the EGZ now move to 1.5 base pair. That there is a problem, because the DNA is supposed to be at the top. The DNA is not supposed to move, because it is still DNA, not amplified DNA. Nothing has been done to it. I have not used any restriction enzyme on you. I have not amplify you. I've not done anything to you. So why are you moving? So if it moves, there's a problem with that particular sample. So your nanodrop and your fluorometer cannot tell you that. Then agarose will come and tell you that, okay, yes, these samples, they are good. So we want to interpret most labs they use nano drop. So we are going to use nano drop to interpret the result now. So like I said, 262 ratio, 1.8 for DNA, 2.0 for RNA, and then 260 to 13. The ideal ratio is 2.0 to 2.2. So this is how the screen of your nanodrop software looks like. You can see the measure, the blank, reblank, print screen, print report, recording, show report, and everything. So when we still have the image of our nano drop. So before you do anything, when you put it on, the machine will bring the software. When you put on your laptop, you click on the software, it's going to tell you to initialize the equipment. Then you are going to do that by putting 2 microliter. That's if your machine is calibrated to 2 microliter. But if it's 1 microliter, you take 1 microliter. So let's say our nano drop now is calibrated to 2 microliters. So I'll take 2 microliter of nucleus free water, not distilled water, not tap water, not running water, not table water, nucleus free water. Then I put 2 microliter on that pedestal. The immediately I close it, the machine will make a sound itself, like it's going to clank, like, and then it's going to read that okay, you have initialized the equipment. The equipment is now ready to be used. Then you open the pedestal again, you clean with a paper towel. Then before you now measure your DNA or your RNA, you have to blank. You want to introduce that sample to the DNA. So the DNA, the machine to the machine to the nano drop. So the nano drop will not be reading water. You want it to know that okay, I want to start measuring my sample now. So let me blank. So you take 2 microliter of any of the sample. No, sorry. You take 2 microliter of your elution buffer, the same buffer that you use to elute your DNA. Sometimes we make mistake with that. We just use any elution buffer, and then we get to when we now get the result, like, oh, what happened? Oh, this. So the same elution buffer you use when we were that same elution buffer we used when we were extracting earlier in the lab. So that same elution buffer will now take 2 microliter of it. So we are now introducing our sample with our elution buffer to the machine now. So you take 2 microliter of that, put it on the pedestal, close it again, then you click on blank. Immediately you click on blank, it will make that sound again. And then it's going to tell you that ready to use. Then you will now take your DNA sample. Before you now take your DNA sample, let's even if we just finish extracting now like this.
No,
Let's
no.
say we finished extracting 751, and then we move to the station where our nano drop is 752. Before I take 2 microliter of my DNA sample, you must vortex. So there's something we call a vortex machine. Is to what's it called, mix sample vigorously, because while that 1 minute journey from the station where we extracted to the station where we want to quantify, the DNA might have settled at the bottom. So we vortex it again. So once we vortex, you take 2 microliter of it, you put it on the pedestal. You will have cleaned the pedestal. For every measurement you do, for everything you do on the pedestal, you clean before you do another thing. You clean with a paper towel. So when you clean it, you put your 2 microliter or whatever calibration your nano drop is, you put it on it, you close it. Then you make sure that the sample type is a DNA. If you are working with RNA, once you click on this drop down, it's going to bring RNA. So you can click on that. Then your sample ID. My sample ID is 8. If your own sample ID is C or whatever sample ID, you put it there. So once you make sure that the informations are, imputed correctly, then you click on measure. Once I click on measure, the nano drop is going to make that sound. Then it starts reading, and then in few seconds it's going to bring this result. So let's say this is my own DNA, the DNA we all extracted earlier. This is my own part now. This is my own DNA now. So this is my DNA. I'm not going to interpret this result. So if you can see, our 260 280 is 1.83, and then our 260 230 is 2.24. So the ratio, and I said 60 280 1.8 for DNA, and for 260 230 2.02 2.2. Now this DNA now is 1.83. So it is very, that means my DNA is very pure. That my 1. In fact, is even above the ideal value. That's 1.8, 1.8 to 1.2.0. So I'm already at 1.83. That means I have high purity DNA with minimal to no protein contamination. So I can use this my DNA now for my PCR. I can use it for uh, my qPCR as well. I can use it for my whole genome studies. So this DNA is good. Then now the 260 is 2.24, and you know the ID value is 2.20 to 2.2. So for it to be 2.2 to 2.24 now, that means this ratio, like there's still some contamination by the organic compounds. Maybe I have some phenol. So it's not that I cannot still use it. It is excellent and is slightly above average. Can you still hear me?
Yes, we can.
Okay, good. So my 260 230 is 2.24. And you know the ID value is 2.0 to 2.2. So when I have this 2.24, so that means there are still maybe there's still phenol in my, what's it called, in my sample. This can still pass. Can, it's just was 0.4 that's been added to it. So it might just be slight instrumental or buffer variability. Now see the 1 gram per microliter. That is the concentration. This 260 230 is the yield. Now this is now my concentration. My concentration is now 65.5. When you extract your DNA, your concentration ranges from 50 to 100, even 150 sometimes. 50 nanogram per microliter to 100 to 150 nanogram per microliter. With RNA, you see this 65.5 that I have. Even if all of these things, the 260, 280, 260 are good, this 65.5 RNA cannot go anywhere, because the concentration for your RNA must ranges from that. You will say that okay, you have enough RNA from 200. Even the 200 you can manage it, you will still be limited. From 300 nanogram per microliter to 1,000 nanogram. If you have 700 nanogram per microliter when you extract your RNA, there's nothing you can use the RNA to do. But even if you have that and your 60 and your 260 280 and 260 230, it's still, there are sometimes you even have minus. That means there's still something wrong. You have the yield, you have enough RNA, but it's not. So what's the essence? What's the essence of having enough RNA and I cannot? It's still impure. It's still impure. Do you understand? So that is, so for this 65, since I'm within the range of 50 to 100, I have 65.5 nanogram per microliter. So I can use this my result
Adedolapo,
for
so
my.
I think your screen, we are not on the same page. We are still seeing the screen that is showing results for nano drops. So we can't see the
Yes, yes, that's
65.
where I am.
Okay, I just wanted to be sure.
Yes, I'm still explaining the the
Okay.
nanogram part. You can see the diagram, right? The graph.
Okay, okay, on the graph. All right. I get it now.
Yes, yeah, it's on the graph. Yeah, 65 nanogram per microliter.
All right.
Yes, yes. So that's how you interpret your result. So this is a good graph. If you, in fact, before you even look at your 260, 280, your yield, your concentration, the graph, once the result comes out, if you see your graph that is, let's say zigzag, just know that something is wrong already. Before you now, go ahead to check what is wrong. So for the method, so we have the nano drop, the qubit, and the gel. The accuracy is moderate, the qubit is high, gel, since gel can tell us about the what's it called, the integrity, not the concentration, and the yield, and the if there are impurities, sensitivity, qubit is high. The cost nano drop is expensive, and qubit is more expensive than nano drop. But gel, just get your agarose powder and prepare your gel. You are very good to go. So that is the comparison of the method. But for a good research work, it's advisable when you do your nano drop or you use qubit, just run it on gel as well to just double check whatever it is that you did. And then best practices, you have to make sure that you use clean and RNA free tubes or DNA free tubes. You calibrate your instrument, you clean that instrument, you make sure you clean every now and then. Once you do your sample one, you clean again, sample two, to avoid cross contamination. And then when you are picking your sample, you have to avoid bubbles. That's where your pipetting skill is, very important when it comes to this as well. And then, like I said, you have to mix your samples properly. So you not just be taking just water. You have to make sure that you vortex it to make sure that, okay, my DNA is not sitting at the bottom. So for quantification, accurate the summary now. Accurate quantification means reliable experiment. And you choose method based on, if I just want to do PCR now, once I use my nano drop and my gel, I'm good. I don't need to use qubit for my what's it called, for to check again for my quantification. But let's say I want to do transcriptomics now, I have to use the three if I have it. But if I don't have the three, I can still make use of the nano drop and uh the what's it called, the gel. Or if it's qubits that you guys that you have available, you can use your qubit and your gel. But the gel is always important, to do a visual check as well, that okay, yeah, the DNA is not moving, it's at where it's supposed to be. And the integrity, yes, I have a very sharp band. So we have come to the end of our DNA and RNA quantification. Okay, so this is how the column looks like. It is attached to a collection tube. So there is silica in between those two. That's where the DNA or your RNA is going to bind to. So you put your wash buffer, elution buffer, or whatever reagent you are asked to inside. Then when you spin it, the flow through goes into the collection tube. Then you can discard if asked to.